Okay, welcome to class, everybody. Because we have a snow day and we were all sent home at 11 a.m., we did not get to have our 12:30 p.m. microbiology lecture. So here we are. I am doing a screen capture video for you guys, and hopefully this will record my voice. I've done a couple of tests and it seemed to work okay. Anyhow, the way I will run this lecture is that you'll be looking at my PowerPoint slides, and I'm keeping it like this because it lets me toggle just through Command Tab back and forth to between the PowerPoint and just a Word document where I'll just write down notes for you. So this Word document over here, this is going to function as our board. Okay, you know I talk, I point to stuff on the PowerPoint, and then I write things on the board. So this Word document will just be our board, and I'll toggle back and forth, but I'm going to leave the slides like this instead of going to full screen. Anyhow, we left off starting to talk about parts of the prokaryotic cell. So how we define the cell depends on having a cytoplasmic membrane. It keeps the outsides out and the insides in. So as we're looking at this over here, because this is a figure, I'm not getting a pointer on this, but here we have our cytoplasmic membrane, and that next layer out, trying to follow it a little bit better, is the peptidoglycan layer, the cell wall. And out here we have an outer membrane. So here's where I would ask you all, if you were here in my living room hanging out looking at the snow with me, whether or not this is a gram-negative or a gram-positive cell. But I will tell you it is a gram-negative cell because you've got this outer membrane right here. This outer membrane is a gram-negative cell. All right. But we're talking about the inner membrane, the cell membrane, which is a phospholipid bilayer right here. And we already talked about how the inner layer and the outer layer in a prokaryotic cell is symmetrical, meaning the fatty acids in this layer are the same as the fatty acids in the outer layer. Proteins are embedded in the cell membrane just as they are for a eukaryotic cell. A lot of these proteins are transport proteins, let things in and out of the cell. Sometimes they are cell communication proteins used for signal transduction so that bacterial cells can communicate with each other. They actually do that. Let's see, but basically the purpose of the cytoplasmic membrane, keeping the outsides out, keeping the insides in. This next bullet point, the second bullet here, is important. That the cytoplasmic membrane plays a role in producing energy for a prokaryotic cell. That is to say that the electron transport system, if a prokaryotic cell has an electron transport system and does cellular respiration, that electron transport system is going to be embedded in the cell membrane because they don't have a mitochondria like eukaryotes do. Eukaryotic cells have their electron transport system embedded in the inner membrane of the mitochondria. But we're talking prokaryotic cells, they don't have that. So, oops, I see a typo here. Oh, no, that's not a typo, that's fine. All right, so, and transport of molecules across the cell membrane. So, here we go. Transport across the membrane can be passive or active. Let's write a note about this. Transport across cell membrane. Active, requires energy. Passive, doesn't require energy. Oh, lucky you guys get to watch me type. All right. Examples of passive include diffusion, which is just movement of a molecule from an area where it is highly concentrated to an area where it is less concentrated. Passive, it happens passively, no energy input is required. Active, an example of this would be, group translocation requires energy. Actually, we're going to get to a slide there later. Okay, passive, such as diffusion, doesn't require the input of energy. So let's say no energy required because it moves a molecule down a gradient, down a concentration gradient. I cannot talk and type at the same time. Apologies, but we're just going to have to deal with this. In fact, passive transport eases a gradient, or it equalizes a gradient. Eases or equalizes a gradient. Active transport requires energy because it moves molecules against a gradient. So it's like concentrating molecules. It takes energy to concentrate molecules, and you guys can't see me making motions at my laptop for concentrating, but going from an area of low concentrated to where a molecule is of high concentration, that would be active transport, and that costs energy. So an example of this, the group translocation, expelling waste products, and the like. Okay, but diffusion, movement of a molecule from an area of high concentration to an area where it is less concentrated. It's like going downhill for small molecules. If diffusion is occurring through a membrane protein, we would call that facilitative diffusion. The protein is facilitating the diffusion, but it is still diffusion because it is still moving a molecule from an area where it is highly concentrated to where it is less concentrated. All right. Active transport requires energy, usually in the form of ATP or proton motive force, because you are making a gradient worse. You're moving it against a concentration gradient. I'm going back one slide here. It would be like this in reverse. Like, say these molecules here are waste products that the cell needs to expel. If the cell takes this molecule and moves it over here, it's going to make this area even more concentrated. So it's moving against the concentration. But sometimes a cell has to do that, and when a cell wants to do that, move molecules against its own concentration, then it costs energy in the form of ATP or a proton motive force. Now, I bet the burning question for all of you guys is, what is a proton motive force? Let's talk about that. Okay, a proton motive force, PMF. Motive force is simply movement of protons across a membrane to generate energy. Movement of protons, movement of protons with a gradient, my bad. Movement of protons with, or let's say down, a gradient to equalize a gradient of protons in order to release energy. Okay, you guys all saw this in earlier bio classes when you studied respiration. This occurs during respiration. In the mitochondria in eukaryotes. But it doesn't have to have, the flow of those protons across a membrane doesn't have to be the inner membrane of the mitochondria. In prokaryotes, you often get protons flowing across the cytoplasmic membrane, generating energy that way. And if you hadn't picked up on this, by the way, a hydrogen ion is what we call a proton because when the hydrogen atom loses that electron, there's nothing in there but a proton left. All right, so the flow of those protons, for example, across the inner membrane of the mitochondria during chemiosmosis, and that releasing energy and allows ATP synthase to pop that phosphate group back onto ADP to form ATP. That whole thing's called chemiosmosis. This should be ringing a lot of bells for you guys. The flow of those protons is actually called a proton motive force. It's just that in respiration, that proton motive force, the energy that's released from that proton motive force, is used by ATP synthase. Proton motive forces can power other proteins, for example, and we'll see an example of that in a little bit. So I just wanted to clear that up. All right. All right, talked about group translocation. This is when a molecule is chemically changed as it passes through a membrane. All right, sorry, I thought I heard somebody at my door and I didn't want to get interrupted by a doorbell. It looks like that was just the neighbors. Sorry, we might get interrupted here. You never know, my dog might start barking. Anyhow, active transport. We are back to active transport. Group translocation is an example of active transport. Wanted to talk about we're talking about that later. Okay, my bad. Sorry, I got thrown off by my neighbor's car arriving. Structures on the outside of prokaryotic cells. You can have a capsule or a slime layer. They are made of the same stuff. I'm not sure where the line is drawn between calling something a capsule is compared to calling it a slime layer, but a slime layer is much more diffuse and a capsule is much more compact. But at some point, we would call a capsule, if it got too diffuse, it would be a slime layer. And I have an example up here, Streptococcus pneumoniae, for example. When it does form a capsule, it does not always form a capsule, but when it does, it causes disease more readily, and we would say that it is pathogenic. So capsules, slime layers have something to do with attachment. They can let a microbe attach to your cells more rapidly. Okay, I think I just heard a snowplow go by or a salt truck. All right. Also on the outside of a prokaryotic cell is a flagellum, or flagella would be the plural. It is a protein structure that's used for movement. It is powered by a proton motive force. So that's what that PMF stands for. A cell could have just one of them. This E. coli cell over here that looks like it's hairy, those are all flagella. We don't see them with our regular basic stains in the laboratory because the proteins just don't absorb. They're not negatively charged to attract those basic stains. You need a special stain for them, and you'll see an example of that, a demo in lab on Thursday. But cells can have one, can have two, can have a lot. Here are some examples. Here we go, monotrichous, the long one right there. This one cracks me up, or it's like a fusilli pasta spirochete with one on each end. This is E. coli right here with several flagella. These ones on the end like this, it looks like, I don't know, it looks like it's a hydra or something, but it's not. Those are just flagella. Okay, pili are shorter versions of flagella, basically. Okay, I have here on the slide, you guys can read it. You don't probably need to hear me read it, but it's used for adherence of cells to another surface, used for some movement, sometimes on a solid surface, but most importantly, in this thing called bacterial conjugation, right there. Conjugation, we need to define. Let me get out of that. There we go. Bacterial conjugation. Bacterial conjugation is exchange of DNA from one bacterial cell to another through what is called the F. pilus. So transfer of DNA from one bacterial cell to another via the F. pilus. The F pilus is a special pilus. The F pilus, it's a longer than typical bacterial pilus. Sometimes it's called the fertility pilus. Or sometimes it's called the sex pilus. It's as sexy as bacterial cells get. Conjugation. It is here, and here's where I really wish I could draw on the board, but I can't. But bacteria have DNA in them, which you know. They have chromosomal DNA, which is a large circular piece. Large circular piece of DNA. And it contains genes that are, the bacterial chromosome will contain genes that are necessary for the bacterial cell to survive. So they produce proteins that are necessary for bacterial cell survival. I mistyped bacterial, too bad. Bacteria also have a different kind of DNA. In addition to the chromosome. And this different kind of DNA is called plasmid DNA. Dun dun dun. Let's give that an exclamation point. Plasmid DNA are small, plasmids are small circular pieces of DNA. In prokaryotes. Eukaryotes, we don't have plasmids. All right, this is bugging me. Here we go. Fix that word there. All right, plasmid DNA. Put that in bold just because I can. All right, plasmids contain genes that are not necessary for bacterial cell survival, but they are genes that will give bacteria enhanced traits. So, genes on plasmids provide bacteria with enhanced traits. Enhanced traits such as antibiotic resistance, So, if a bacterial cell has a plasmid on it that allows it to make the F pilus, it can have contact between, it can contact another bacterial cell that doesn't have that plasmid, and that a copy of that plasmid will travel through the F pilus. That F pilus will connect to bacterial cells, and they can exchange plasmid DNA between them. If those plasmids provide genes that will allow that bacterial cell to resist the effects of, say, penicillin, it can pass that trait on to a whole other bacterial cell that didn't previously have that trait. So one of the main ways that bacterial cells can acquire antibiotic resistance is through conjugation through the F pilus and exchanging DNA with other bacterial cells. In lab, in about a month or so, we will do a conjugation experiment. but for now, you need to know bacterial conjugation is the transfer of DNA from one bacterial cell to another via the F. pillus. So that's an important one. Here we go. Otherwise, inside a prokaryotic cell, they have DNA. They have ribosomes that are 70S ribosomes. I'm going to add that right there. You got a couple of storage granules, a little picture here for you, and I promised last week I'd show you a stain of an endospore. Here you go. Just please, for right now, know that endospores are a dormant cell type, and they are heat resistant. The only way to destroy cells that have formed endospores is to use high pressure and much higher temperature. All right, so we already talked about prokaryotic DNA, chromosomes, supercoiled DNA containing the main genes necessary for survival. Plasmids are circular DNA with a few genes providing enhanced traits such as antibiotic resistance. All right, let's talk about ribosomes. Ribosomes are what do translation. They look at a piece of RNA and they will build a protein based on what those G's, A's, U's, and C's say. Ribosomes are made up of two subunits. All right, the bacterial ribosome is a 70S ribosome, and it is made up of a smaller and a larger ribosomal subunit. In bacteria, the 30S, the smaller ribosomal subunit is size 30. The larger one is size 50. Yes, I know it does not add up. 30 plus 50 is 80, but it doesn't add up like that. That S actually stands for Svedberg, the dude who invented the centrifuge, and these 30S and 50S sizes have to do with, for example, how this 30S subunit will travel in a medium in a test tube when you put it into a centrifuge. The 50S will get to the bottom of the test tube in a centrifuge faster than the 30S. But when you put them together, the whole piece does not move to as they would if they were an 80. They move as they would for a 70. So that's what that has to do with. So if you're ever on Jeopardy, you can say, I know what that S stands for. All right. But bacteria have a 50S ribosomal subunit and a 30S, whereas eukaryotes are a little bit different. So ribosomes. The 30S ribosomal subunit in bacteria and prokaryotes plus the larger one, the 50S, gives you a 70S sized ribosome, and that's prokaryotes. In eukaryotes, the smaller ribosomal subunit is a 40S, and the larger is a 60S ribosomal subunit, and that gives you an 80S in eukaryotes up here. Prokaryotes, there we go. All right, so that's another difference between prokaryotes and eukaryotes. Remember that list that you guys have? Way back here, I'm going to have to get out of this, see if I can, here it is, slide 21. Here we go. We got a difference in ribosomes, 80S ribosomes versus 70S ribosomes, and it's one of the, one class of antibiotics will target 70S ribosomes as opposed to 80S ribosomes. You guys have a few more things to add here onto this list. Remember I asked you to put it on like the inside back cover of your notebook so that you can add to it throughout the semester. Okay, we're out of room on this slide. So I'm going to go back to the slide where I was at. Where was I at here? Transport across the membrane. Prokaryotic DNA. There we are, ribosomes. All right, so let's write this down. We have a few more things that we need to add to that prokaryotes versus eukaryotes list. Prokaryotes, I don't want to divide this up. We're just going to have to keep it like this. One of the things that we can add is that they have a symmetrical cell membrane, whereas eukaryotes have an asymmetrical cell membrane. They have different size ribosomes. That's already on the list. But another thing for prokaryotes, you know what? I'm going to do it this way. Do it as a bulleted list. Here we go. Prokaryotes also have, let's see, cell membrane plays a role in producing energy. That was on a previous slide that you can add to your list. So for example, the electron transport system is embedded in cell membrane. The bacterial flagellum. Bacterial flagella are powered by a proton motive force. That's different from eukaryotes. So let's do this as a bulleted list too. Okay, in eukaryotes, the cell membrane plays no role in producing energy for the cell. And the eukaryotic flagella, we're going to see this on a later slide, but I'll just give it to you now. Oops. Are powered by ATP, not directly by a proton motive force. So there we go. Those are some things to add to that list. I should also note that ribosomes are made up of ribosomal RNA. So let's get back up to here too. Ribosomes. Made of ribosomal RNA. That little r stands for ribosomal. And actually, the two ribosomal subunits, the 30S and the 50S, those are made up of even smaller pieces of RNA. Like there's a 16S RNA. And those smaller pieces of RNA those were sequenced by that dude, Carl Woese, and he used that data to show relatedness and to construct the 3-domain system that we use. So basically he was looking at ribosomes. So what that basically says is that organisms that have similar sequences to their ribosomal RNAs are very related to each other. That shows that is a bit better predictor of relatedness than anything else we've looked at before. So ribosomes, they're what's for breakfast. No, they're all about making proteins. All right, so let's get to the structures of a eukaryotic cell that we don't see in prokaryotes. All right, the plasma membrane, yes, the eukaryotes and prokaryotes both have a plasma membrane, but the bilayer is asymmetrical and there is no role in energy production in the eukaryotic cell membrane. All right, transport across the membrane. I wanted to talk a little bit about that in eukaryotes. Okay, protein channels will let small molecules in and out as they do for a prokaryotic cell, but with eukaryotic cells you get endocytosis and exocytosis. So endo right here means to come in to something, whereas so bringing in a substance, where exocytosis is secreting a substance. So exo meaning to go out from. There are two types of endocytosis. Pinocytosis up here, particle is brought in. These would be for particles that are in solution. So a particle in solution is brought in. So it's brought in, and we call it an endosome. I'll show you in just a little diagram of that. An endosome would just be part of the cell membrane. Those phospholipids will form a little bubble or a vesicle around that. And we call it an endosome. Really, it's a vesicle. That's it. Phagocytosis or phagocytosis, however you want to pronounce it, that's when the cell forms those pseudopods, those false feet, to grab a larger particle that is not in solution. So some people think of pinocytosis as cellular drinking and phagocytosis as cellular eating. It's not entirely accurate, but it's one way to remember. But let's take a look at what it looks like. Okay, so pinocytosis, small particles that are in solution. The cell membrane would pinch itself off and basically form a bubble around it. If it's fused with a lysosome is just basically a little vesicle with digestive enzymes that will break down the foodstuffs. Likewise, phagocytosis, these pseudopods, these false arms would form. There is one class of protozoa that does that a lot, and we'll look at that one. It would bring in a particle using pseudopods, form a vesicle or an endosome around it. It would fuse with a lysosome, a vesicle containing digestive enzymes that will break down the foods. Okay, over here we have a vesicle where, say, a protein that was formed in the rough endoplasmic reticulum and modified in the Golgi apparatus would be packaged up in a vesicle. That vesicle would actually fuse with the cell membrane and spit out the substance, the protein, for example, outside the cell. You don't get endocytosis and exocytosis in prokaryotes. All right, so secretion of a protein, something synthesized by the ribosome in the rough endoplasmic reticulum. Eukaryotic cells also have cytoskeleton. Prokaryotes don't. Cytoskeleton network of thread-like proteins that will change, that can disassemble itself and reassemble itself into a different form in order to meet the cell's needs. One of the time changes to meet the cell's needs, like when would the cell have a need for having a slightly different shape? During mitosis is a big example of that. All that stuff where the mitotic spindle forms, those microtubules of the spindle are part of the cellular cytoskeleton. So here we go. Here's better than what I could draw. We have our microtubules, our intermediate filaments, and these actin filaments. They form a mesh-like network of proteins within the cell to support everything. So you don't have, like this mitochondria swimming around in the cell and bumping into other things. It holds things in place, gives structure to the cell. I liken them to those K'NEX toys, K''NEX, where you can build a dinosaur or a truck or the Eiffel Tower or a Ferris wheel out of it. And then, after you build it, you can take it all apart and rebuild it again. So kind of the same concept. All right, the eukaryotic cell. Eukaryotic cells have flagella. Eukaryotic flagellum would be used for motion. But the shorter version of flagella are not called pili in eukaryotic cells. We call them cilia. Cilia are short versions of flagella. So let's actually write that down. Okay, so let's see. Eukaryotic cells have cilia, not pili. And prokaryotic cells have pili, not cilia. We'll get back to this in a second. Let me show you a picture here, okay? They are both made out of protein in what's called a 9+2 arrangement of microtubules. Here we have 1, 2, 3, 4, 5, 6, 7, 8, 9 sets, and then 2 separate sets in the center. So that's called a 9+2 microtubule arrangement. But do you see this little covering here? That is actually the cell membrane covering a bacterial flagellum. So here is a major difference between eukaryotic flagellum and cilia compared to prokaryotic flagella and pili. Eukaryotic flagella and cilia are both covered by the cytoplasmic membrane, so they are technically inside the cell, not the case with prokaryotes. So that's another difference between prokaryotes and eukaryotes that you need to know. So with prokaryotic cells, flagella and pili are truly outside the cell. They are not covered by the cell membrane. Not the case with eukaryotes. Flagella and cilia are covered by extensions of the cell membrane. So they are technically inside the cell. So this stuff is all in addition to what you have on that slide and in your notes previously. Alrighty, let's get into the nucleus. Okay, and I know you guys already know that the eukaryotes have nuclei, and that there is supercoiled DNA with round around histone proteins in there. But I wanted to point out to you guys this little purple spot. I'm thinking back to Bio 100. You guys in lab had some cell models you had to look at, and I think the nucleus was green on the outside and purple on the inside, but this purple spot is called the nucleolus. That is where ribosomal RNA is made and produced, and I do want you to know that. DNA is supercoiled with histone proteins. That's already on your prokaryotes versus eukaryotes list, but let me show you what they look like. Okay, so these little blue disc-like looking things, I don't know, they look like blue bonbons or something. Those are to represent histone proteins, and the red ribbon as DNA. This is an electron micrograph of chromosomal DNA, eukaryotic, and these little grainy dots are actually the histone proteins. That is really amazing that we can actually visualize that. Really cool. All right. Eukaryotic cells have mitochondria. You guys know that they have an inner membrane with the cristae, an outer membrane that is smooth, but inside, if I make this a little bit bigger, going like this. Oh, what the heck. There we go. There is actually the space in between. Inside the cristae is called the matrix. Inside the matrix there are ribosomes and mitochondrial DNA. So let's write that down. This really isn't a difference between prokaryotes and eukaryotes. So I know you guys already know that prokaryotes don't have mitochondria, but regarding the eukaryotic, well, that's redundant. Regarding the mitochondria, the matrix contains mitoDNA and 70S ribosomes. So before you go all what the frack on me on this, saying I thought eukaryotes only had 80S ribosomes inside the eukaryotic mitochondria, there are 70S ribosomes. So if we take a look at this mitochondria right in here, somebody a long time ago was like, huh, that's kind of funny. I just got a text. Go away. I just lost my train of thought. Okay, somebody at some point was like, huh, look at this mitochondria. It looks like it's about the size of a prokaryotic cell. I wonder what's inside of it. Let's look at what's inside the mitochondria. There's DNA in there. Let's sequence this DNA. The DNA of mitochondria matches up to prokaryotic DNA. And the ribosomes inside the mitochondria match up to the size of ribosomes inside prokaryotic cells. The idea is that the modern day mitochondria descended from a long ago ingested bacterial cell. That theory is called endosymbiosis. So endosymbiosis is the idea. Let's just go on to another page here. There we go. Endosymbiosis is the theory that the modern mitochondria is the descendant of an ingested bacterial cell. And the evidence that we have for this is that it's about the same size as a prokaryotic cell, same type of ribosomes, 70S, and also mitochondrial DNA matches prokaryotic DNA. Just kind of general. We get some general matches there. All right, now when it comes to the chromosome, when it comes to the chloroplast, okay, we got this. It was actually the chloroplast that led to the endosymbiosis theory being developed, because similar to a mitochondria, the chloroplast is an outer membrane and an inner membrane. We call this the folds on the inner membrane the thylakoids, not cristae. And we don't call the area in between the two, the matrix, we call it the stroma. But inside the stroma, so chloroplast, chloroplast, inside the chloroplast, inside the stroma are 70S ribosomes. And chloroplast DNA, I'm just going to write chloro-DNA. Somebody, and I know who it was, the woman named Lynn Margulis, I think I spelled her last name. Lynn Margulis was a scientist at UMass. Now, she did not do this work at UMass. I believe she did it when she was at Cornell. Developed the endosymbiotic theory. She developed the theory of endosymbiosis, working with chloroplasts. And she postulated, well, okay, gee, chloroplast inside the plant, where's my bullet? There we go. Chloroplasts are about the same size as photosynthetic bacterial cells. So they're about the same size. She's like, and they also have 70S ribosomes. She's like, well, dang, we gotta sequence chloroplast DNA. Let's just write chloro-DNA. And she found that chloroplast DNA matches not just prokaryotic DNA, but it strongly matches, let's put this in there, strongly matches the DNA from a photosynthetic prokaryotic cell that is very sophisticated. And this cell is still around today, and it is cyanobacteria. They used to be called blue-green algae, and they are still around today. So we refer to cyanobacteria as probably the most sophisticated prokaryotic cell that is in existence today. Cyanobacteria, they are prokaryotes. They do not have mitochondria. They don't have their chromosome inside a nucleus. They are definitely prokaryotic, but they're very sophisticated. They are more sophisticated than other photosynthetic bacteria that only have one or the other photosystems. Cyanobacteria actually have both photosystems. And that's just really cool because not only does their DNA match that of the chloroplast, cyanobacteria are still around. So they are the bomb. They used to be called blue-green algae. So there we go. So Lynn Margulis, endosymbiosis. You can Google her if you like. She was UMass's, I think, only member of the National Academy of Sciences. And she died about 10 years ago, but she actually gave a talk at HCC about 10 years ago. Fascinating lady. Fascinating scientist. Okay. All right, we talked about the endoplasmic reticulum. The rough endoplasmic reticulum is only in eukaryotic cells. It's called rough because it has ADS ribosomes attached to it. It is all about producing proteins, whereas the smooth endoplasmic reticulum does not have any ribosomes on it. That's why it's called smooth. And it has nothing to do with proteins. It only breaks down lipids. The Golgi, getting ahead of myself. Okay, so here we go. Electron micrograph of rough endoplasmic reticulum. That's what all that grainy stuff is for. The squiggly looking stuff down here is the smooth endoplasmic reticulum. It's usually involved in lipid synthesis and breakdown, and sometimes detoxification of drug molecules. Here we go. The Golgi apparatus receives proteins from the rough endoplasmic reticulum and modifies them. So, for example, it might add a polysaccharide or a short saccharide molecule to a protein, or it might add a lipid molecule to a protein. So you'd have a lipoprotein, and it would package it up into a vesicle and, for example, send it elsewhere in the cell or outside the cell. All right, and that's it. So we are all caught up. We will meet on Thursday, and we'll take it from there. You will have a reading assignment to do, and I hope we don't have to do this again. So I'll see you guys in class on Thursday. Take it easy.